Hi, my name is Kelly Giffey, and I am a senior at WFS High School in Chehalis, Washington. Last year, I conducted my research project on the correlation between dietary intake and agouti-related protein expression levels in zebrafish located in the hypothalamus. When I was coming up with ideas and topics for my research project last year, I wanted to have a topic that had relevance and was important in today's country. Today, obesity has become a problem. It has overtaken smoking as the #1 cause of death because it leads to so many other complications such as diabetes, heart disease, and many other complications. I found a gene that is related to body weight, and that's the agouti-related protein. And I wanted to see how this mechanism works. So before meals, you produce ghrelin in the lining of your stomach called the fundus. That ghrelin, that hormone, goes up to the brain, binds to a receptor, and signals to your body that you're hungry because it produces AGRP in the brain. As you intake food, you secrete leptin, which is an antagonist of ghrelin, and signals to the body that you are full because it binds to the receptor in the brain, the same receptor, and signals, tells your brain to stop producing that protein, signaling that you're satisfied. So I wanted to conduct a research experiment on this. Zebrafish have the same genes, or they have the similar organs as humans do, and they also contain this protein, and it is expressed in their hypothalamus as well. And in order to view the gene expression, you need to utilize real-time PCR. And in order to use real-time PCR, you need a tissue sample, so zebrafish were essential in this experiment. I set up 2 tanks. One was the control tank, and one was the experimental tank. 15 fish were in each tank. Each fish weighed about 0.5 grams. And the control tank was fed 4% of their body weight, which is normal, and the other fish were fed 2% of their body weight. Before this experiment took place, I hypothesized that the fish that were fed less would have higher and elevated levels of AGRP expression in their brain because they were intaking less food, so they would be secreting less leptin to halt AGRP reduction. They would also be producing more ghrelin in the lining of their stomach because they would be more hungry before meals. This study lasted over 9 days, and I have the calculations I made for the daily feeding charts that I fed each tank. Once those 9 days were over, I euthanized the fish and dissected their brain tissue out of their fish. The brain tissue was placed into an EpiTube where I extracted their mRNA from that brain tissue sample. That mRNA was then converted into cDNA through the process called reverse transcription. mRNA is single-stranded. During reverse transcription, they bring in free-floating nucleotides using PCR and produces multiple copies of that mRNA. Once the DNA was ready to use, it was all double-stranded, I could use it in real-time PCR. After I used it in real-time PCR, these were my results. As you can see, this is the 18S control right here. 18S is a good housekeeping gene to use because it is located in every cell. It's always turned on because ribosomes are found in every cell, and that's what it codes for the 18S ribosomal subunit. This was the AGRP expression right here. I unfortunately was unable to conclude very little about AGRP expression in the brain due to the fact that it is located in the hypothalamus, which is a very small region of the brain. It's in that circle, the green circle right there. It's about 1/100th of the size of the brain. I could conclude that it has very low transcript level in comparison to the entire brain, but if I was able to look specifically at that hypothalamus, I could probably get more conclusive results, or perhaps use tissue cell culture and try to see if I could grow cell lining of just the hypothalamus cells and see if that could give me more conclusive results. I'd like to thank WFS High School and my teacher, Mr. Henry Weeks, for helping me with this project. Thank you so much for your time. Bye.